This video will cover the enzyme-linked immunosorbent assay, or ELISA, a topic important for various competitive exams, including CSIR-NET, UGC NET, ICMR-JRF, DBT exam, NEET, and post-medical and non-medical interviews. ELISA is an immunological technique developed in 1971 by Peter Perlman and Eva Engvall. It detects and quantifies antigens or antibodies using enzyme-labeled antibodies and a color-producing substrate. ELISA is preferred over radioimmunoassay, RIA, as it avoids radioactive materials. ELISA is based on specific antigen-antibody interaction. The steps involve immobilizing an antigen or antibody on a microtiter plate, binding a specific antibody or antigen, adding an enzyme-linked antibody, and adding a substrate. The enzyme converts the substrate into a colored product, and color intensity is proportional to the concentration of the antigen or antibody. Key requirements for ELISA include the antigen, antibody, and enzyme. Commonly used enzymes are horseradish peroxidase, HRP, and alkaline phosphatase, ALP. Substrates like TMB and OPD are used, along with a microtiter plate and a stop solution. Direct ELISA is one of the 4 types. The principle involves coating the antigen on the plate, followed by direct binding of the enzyme-linked primary antibody. Advantages include fast and few steps, but disadvantages are low sensitivity and no signal amplification. Indirect ELISA principle involves coating the antigen, followed by the primary antibody binding to the antigen, and then the enzyme-linked secondary antibody binding to the primary antibody. Advantages are high sensitivity and signal amplification, while disadvantages include cross-reaction and more steps. Sandwich ELISA involves coating a capture antibody, which binds the antigen from the sample. A detection antibody then binds the antigen. This type offers very high specificity and sensitivity, but it is expensive and requires a matched antibody pair. Competitive ELISA involves the sample antigen competing with a labeled antigen for binding to the coded antibody. The most important concept here is that color intensity is inversely proportional to the antigen concentration. This method is advantageous for detecting small amounts of antigen, but complex result interpretation is a drawback. ELISA has diverse applications. In medicine, it is used for detecting diseases like HIV, hepatitis B, dengue, and COVID-19 antibodies, as well as pregnancy testing and hormone analysis. In research, it is used for cytokine measurement, vaccine development, and protein quantification. In the food industry, it detects allergens and toxins. Blocking agents such as BSA, casein, and gelatin prevent non-specific binding. The washing step removes unbound antibodies or antigens, commonly using PBS tween buffer. Detection methods include colorimetric, the most common, fluorometric, and chemiluminescent. Absorbance is most commonly measured at 450 nanometers.